Hi, I'm John Kiriakou, and welcome back to Deep Focus. When I was a little boy growing up in Pennsylvania, we had thousands and thousands of chestnut trees in our area. We used to throw the chestnuts at each other for fun, and our parents would gather them and roast them as a snack. But then disease struck the American chestnut population. I haven't seen a chestnut tree in decades. They're almost all gone. But scientists now say that they've come up with a plan to revive the American chestnut through genetic engineering. To some, that may sound like a great idea, but is it really? It's never been done before, and we have literally no idea what it will do to other trees as its pollen is spread by the winds, by bees, and by other insects. Are we saving the chestnut, or are we dooming other tree species? Scientists have been working for years to revive the American chestnut, a large tree that once covered the eastern part of the country and provided food for humans and animals alike. The tree usually lived for hundreds of years and made up an important part of the Eastern American tree canopy. But beginning in the early 20th century, the tree was affected by something called chestnut blight. That's a fungal disease that came from imported Japanese chestnuts. Beginning in 1904, and spanning the 20th century, it's estimated that the blight killed between 4 and 6 billion American chestnut trees. Now mainly stumps and root systems remain. They produce young saplings, which usually at some point just become infected with chestnut blight and then die. There are, however, an estimated 4 million American chestnut trees still growing out in eastern U.S. forests, including some that are mature and that did not die from the blight. Several groups of scientists are working to revive the American chestnut, including a group at the State University of New York College of Environmental Science and Forestry. Those scientists had the idea to insert something called the oxalate oxidase gene from wheat into the DNA of the American chestnut to neutralize the oxalic acid produced by chestnut blight that was responsible for killing the trees. In 2020, these scientists asked the government for permission to introduce the new genetically modified trees into the wild. That sounds great, right? Well, no, actually, it's not great. First, we have no idea what the environmental impact will be of introducing a genetically modified tree into nature. It's never been done before. And even if we had an idea of what would happen, we still don't know what will happen, say, 50, 100, even 200 years from now. Remember, these trees live for centuries. And perhaps most disturbingly, these same scientists made a very serious mistake. Instead of using pollen from the genetically engineered chestnuts known as D58 in their experiments, as far back as 2016, they were accidentally using pollen from a different genetically engineered chestnut called D54, which has the OXO gene in a different part of its genome. As a result, an important gene was deleted, causing the genetically engineered trees to become sickly. Many lost their resistance to the blight and died. Almost all of the thousands of test trees are D54s, not the hardier D58s. Again, we have literally no idea what the effect will be if these genetically defective GE trees were released into forests. Well, we're happy to welcome a guest today who is an expert on this important environmental issue. Anne Petermann is executive director of the Global Justice Ecology Project, which she co-founded in 2003. She's also the co-founder and is international coordinator of the campaign to stop GE trees. Anne, welcome to the show. Thanks so much for joining us.
Thanks so much for having me, John.
And let's start with the basis of this problem. Tell us about the American chestnut, about its coverage 100 years ago, about what happened to it, and why its destruction has been almost complete.
Sure. Yeah, it was a major tree in the eastern forests of the United States, especially up the spine of the Appalachians. And some estimate it was about 1 in every 4 trees. People called it the redwood of the East because it was such a large tree and it was, amazingly tall and very rot resistant. So it was used for everything from telephone poles to cribs to coffins to houses to fences. I mean, people used it for everything, which actually was driving in the South, in the southern part of the United States, in those forests, it was actually being over harvested. It was being harvested faster than it could grow back. And then the chestnut blight, as you mentioned, came along in around 1901, imported on Japanese chestnuts that ended up in the New York Botanical Garden. And so that was ground zero for the infection. And then it spread just incredibly fast. People were trying just from the very beginning to figure out what to do about it and try to stop the blight. And they had all kinds of crazy ideas. In the state of Pennsylvania, they cut a swath of forest down that was miles wide across the entire state, thinking they could stop these fungal spores from spreading, but they couldn't, of course. And so what ended up happening was people were encouraged to cut down healthy trees in advance of the blight so that they could at least get the timber out of the trees. So that means what ended up happening was a lot of trees that might have had blight resistance were killed, were cut down on purpose. So between the blight and the people cutting them down to try to save the wood, the trees were largely decimated. But since then, people have continued to try to bring them back, to come up with solutions to the blight problem. And there have been lots of different teams of people doing that. And one of them has been doing it with wild American chestnuts, breeding them together that have some level of blight tolerance, and they've been very successful. And as of 2023, they're starting to release what they believe are self-sustaining, blight-tolerant wild American chestnut trees.
I want to get into well, no pun intended, but the roots of that for a minute. When did I first of all, I'm assuming that work to save the American chestnut did not always include genetic modification. So when did work begin to save the chestnut? What did that work look like? And how is it that it morphed into genetic modification?
The work to save the American chestnut really started almost as soon as people noticed the
Oh,
blight was moving through the forests at such an amazing pace and killing so many trees. And it's included all kinds of different approaches, from crazy spraying of herbicides to uh you know as I mentioned, breeding wild American chestnuts with each other. Some people have been trying to breed Chinese chestnuts, which are naturally resistant to the blight, with American chestnuts, thinking they can get a hybrid that is resistant to the blight. They've gotten actually as far as a 16th wild American chestnut that is pretty resistant to the blight. Unfortunately, it very much has the physical characteristics of a Chinese chestnut, which is not great. It's more of a bushy tree, so it's not great for forests. So they're not thinking that that's going to be something that can help restore the forests. And then I think because the other approaches were taking so long, people were getting antsy. They wanted something faster, and they felt that genetic modification would be a fast way to take care of this problem. So around 1989, they started looking at ways to genetically engineer the chestnut. And that's when the scientists at the Environmental Science and Forestry School in Syracuse started to get involved.
What's the role of government in all this, Anne? Is government a neutral observer here, or is it pushing a solution that involves genetic modification?
Government has, well, in with genetic engineering overall, the USDA, which is the body that oversees genetic engineering organisms in the United States, and the FDA has largely rubber stamped them. There have been a few that have been rejected, but not very many, considering the number that have gone up to the USDA for approval. The U.S. Forest Service was actually involved early on in the whole chestnut genetic engineering scheme, supporting it, and as was Duke Energy and other companies. So the US government has really been a promoter of this, and thinking that it would be a good way to bring back the chestnut and all of the industrial purposes that the chestnut could bring, like timber, like biofuels, biomass for electricity burning, et cetera, et cetera.
I want to ask what may sound like a little bit of a silly question, but is chestnut blight specific just to American chestnuts, or are there other trees like the buckeye tree, for example? I said in the intro, I haven't seen a chestnut tree since I was a little kid. I haven't seen a buckeye tree since I was a little kid, and they used to be everywhere too, growing up in Western Pennsylvania. Um, but are we talking just solely about the American chestnut, or are other trees, other species also affected?
Related species related to the American chestnut definitely have been affected, like the chinkapin, which is further in the southern range of the American chestnut. And some trees, like oak trees, are a carrier of the
Oh, for
I'm
heaven's
sorry, of the
sake.
chestnut blight, but they don't. It doesn't kill them, but it resides on them. The fungus resides on them. But there are, the problem with globalization in this day and age is the global trade in whole logs and wood chips means that we have so many invasive pests and pathogens in our forests. It's incredible. There's just been no effort at all to try to keep the wood chips, clean, make sure that we're not importing all these pests and pathogens or exporting them for that matter. So, a lot of trees are in trouble. The hemlock, had the Willia delgid that's attacking them. I mean, I could go on and on about that. There's just so many.
Sure, Dutch elm
But
disease.
yeah. And so that's one of the things researchers say is if they can successfully genetically engineer the American chestnut, then maybe they can use similar techniques to address some of these other forest pests and pathogens. But the reality is, over 30 years after they started genetically engineering the American chestnut, they had something that they said that was ready to go. They took it to the USDA. They gave them all the research that went along with it. 3 years later, they found out it was genetically defective and dying. So, genetic engineering is by no means an exact science. And because we're talking about trees that live for hundreds of years and that live in wild forest ecosystems that have unbelievably complex interactions with other species, there's really no way that they can know that these things will ever be safe.
Can you explain to us what the process of genetic modification is for trees? It seems like it has to be done in a completely closed environment, which of course is a good thing. You don't want anything to escape from a lab. But then is it all just released into the environment?
It starts out in a lab, yeah, as you mentioned, growing clones in petri dishes and all of that, the kind of thing that you would expect. And then they start growing them in the greenhouse. So they get them started in the greenhouse, obviously a very controlled environment as far as moisture and nutrients and temperature and light and so forth. And then eventually they move them out into controlled field trials. So these are outdoor field trials, but also very controlled conditions. They're not a forest. And then eventually the idea with the American chestnut was after it had been growing in the field for 3 years, the test results looked like they were pretty good. Then they asked the government to give them permission to just release them into the forest completely unmonitored, unregulated, with no idea where they were going to be going. And, going from a 3 year field trial, controlled field trial with a species of plant that can live literally 300 years to releasing it into the forest is after 3 years, it's just crazy. I mean, they have absolutely no idea what would happen to the tree. They didn't bother to wait to see if the tree even became mature. You would think that a scientist would at least want to see a seedling become a mature tree and see what happens to the genetic modification over that time period. But when you're talking about 20 years before the tree is mature and starts producing seeds and nuts, yeah, they didn't want to wait that long. So they said, we've got enough information. This is good. The interesting thing about the tests that they did, the safety tests, they did tests to make sure this was safe for bumblebees, and they did tests to make sure that this was safe for salamanders and other things. They didn't even use the leaves from the Darling 58 trees, or the Darling 54, as it turns out. They didn't even use those leaves. In the case of bumblebees, they didn't even use genetically engineered pollen at all because they didn't have any. So they used regular pollen from chestnut trees, which they mixed with the enzyme, the OXO enzyme, and said, that's the same thing, which is kind of ridiculous, actually.
I want to drill down into that a little bit more. Even if we do study these trees for 10 years or even 20 years, we still have really no idea what the long-term effects will be of releasing them into the environment, as you just said. What is it that we should be afraid of? Let's say scientists take these saplings and plant them in the forest, declare victory, and just go home. What should we be afraid of? Is it that they may come to dominate other species? Is it that they may harm other species or overtake them? Or that these genetically modified chestnuts will just fail?
Yes, all of the above. Um,
Gosh.
with the genetically engineered American chestnut, had the researchers gotten the approval to release these things unmonitored into the environment on the time scale that they wanted, they would have already been growing throughout the forest wherever
Right.
people had planted them before this genetic defect was discovered. And this genetic defect, as you mentioned, causes the trees to get sick. It causes them to lose their blight resistance and die. Many of them just plain old die. That would have meant that these genetically engineered American chestnuts would have been eventually spreading their seeds and pollen throughout the forest, without knowing where they were planted because they were deregulated, right? And
Oh, yeah.
contaminating the wild American chestnuts, what they call the mother chestnuts, contaminating them with this genetically defective pollen, and then on and on and on. And you don't know, I mean, what ends up happening after 20, 30, 100 years? They don't know. We don't know. Nobody knows. But certainly it's not a good thing. And I believe it could have been the thing that, was the death knell for the wild American chestnut, because, here's another thing that comes along that knocks it down.
I hope you don't think this next question is a silly one, and it's a little bit selfish because I'm just interested personally. But I go to Greece a lot. I have family there. I love the place. And every single time I go, there are dozens, if not hundreds, of street vendors selling roasted chestnuts. They'll sell chestnuts and corn and nuts and things like that. And it's not just in Greece. I've seen them in Italy. I've seen them in London. I've seen them in Cyprus. What are those chestnuts? Where do they come from? Are they American chestnuts? Are they Japanese? And how is it that they're able to produce so prolifically?
Those are European chestnuts. So there
Oh, yet
are a
another chestnut.
variety. Yeah, there's different subspecies of chestnut. So there's an Asian chestnut, there's a European chestnut, there's the American chestnut. And yeah, so they grow very well in other places where they haven't been subjected to this blight, or where they have resistance to the blight already. And it was just in the United States and a little bit of Canada, where the American chestnuts were decimated by the blight. So, but you can still, like, if you go to the co-op or the grocery store at the holidays, you can see
Right.
bags of chestnuts that you can buy, and largely those are imported from Europe.
I got it. And tell us about this lab error that occurred back in 2016. The media say that it was such a significant mistake that it set research back by decades. I've actually read that a number of times. What happened exactly? And what is the effect of the mistake? Did it really set research back?
Yes, it definitely set research back. So the researchers had been trying, through trial and error, which is what genetic engineering is. And, well, most science, right? So had been through trial and error. They finally came up with a tree that they thought was going to give them blight resistance without a lot of problems. And that was called the Darling 58, or the D58, named after Herb Darling, who was a construction magnate from Buffalo, New York, who funded a lot of the early research. And so they said the Darling 58 was the way to go. They had brought on board, the researchers at ESF, the SUNY school in Syracuse, had brought on board researchers from the American Chestnut Foundation, which was also trying to restore the chestnut to the forests. They convinced them that genetic engineering was the way to go. So the American Chestnut Foundation was literally giving the ESF researchers millions of dollars, as well as collaborating with them on field trialing the genetically engineered chestnut trees, helping them with research, helping them with the genetic modification process, etc. And what happened back in 2016, and nobody knows exactly what happened with this, the researchers at ESF gave the researchers at the American Chestnut Foundation the wrong pollen. So instead of giving them pollen from the Darling 58 trees, they gave them pollen from the Darling 54 trees, which was a completely different genetically engineered variant, which, as you mentioned, had the engineered gene, the transgene in the wrong chromosome, which deleted an important gene for drought tolerance. So as a result, the trees just are sickly, they die. And the only reason that they figured this out eventually was the trees started dying in 2023, 3 years after they asked the government for permission to plant them out in the forest. The trees started getting sick, they started dying. The researchers couldn't figure it out. Some other researchers from another university actually had the equipment, because ESF didn't have the right equipment, TACF didn't have the right equipment to figure out what was going on. This other university did, so they looked into it very deeply and said, this is not a Darling 58 tree. This is a Darling 54 tree,
Oh my God.
et cetera, et cetera. So, here we have people doing this genetically engineered tree research who don't even have the equipment to understand exactly what they're doing, much less what the impacts will be. So the whole thing has been a real cautionary tale. But because of that pollen mistake, if you will, the American Chestnut Foundation ended up spending years and millions of dollars in research on the wrong tree, as did ESF. So ESF is trying to play it down and say, oh, they're just a little bit different, these D54s and D58s. It doesn't really matter, but it matters a lot. And we still don't know what the Darling 58 trees would do. In fact, one of the scientists that we work with explains that even if they had the Darling 58 trees, and even if this lab error had never happened, the Darling 58 trees, they have what's called a cauliflower mosaic promoter in them, which is how the gene that's inserted is promoted to actually be expressed. And they said that was used on both the Darling 58 and the Darling 54, and it is notorious for what's called gene silencing, which means that the trait that's expressed by the transgene eventually is just shut off by the plant. It just stops working. So even if they had the Darling 58s, they would probably eventually have lost their blight tolerance anyway.
Oh my God. So here we are all these years later, all these millions of dollars later. Do you believe that the American chestnut can be saved? Is this a fight worth fighting?
It can be saved, absolutely. It's just not going to be saved with genetic engineering. That's a red herring. That's a Trojan horse. The biotechnology industry and the timber industry back in the 80s and 90s decided that the American chestnut tree would be a great tree to use as a genetic engineering, as an example of what could happen with genetic engineering of trees. They don't care about bringing back the chestnut. They care about genetically engineering poplars and pines for industrial purposes. That's what they want. And the public has been overwhelmingly opposed to that. And so they needed a charismatic tree that would allow them the ability to open the door to these other industrial trees. So that's what the American chestnut is being genetically engineered for. It's a Trojan horse to open the door to all of these industrial trees. But
So
getting back to the question about, can it come back? Yes,
yeah.
absolutely, it can come back. In fact, it is coming back. So we work with people who are growing American chestnuts, wild American chestnuts, blight tolerant American chestnuts. There's the American Chestnut Cooperators Foundation based out of Virginia. They've been breeding these trees for 60 years. They say they have one that's self-sustaining. We work with a landowner in Maine he started out with 40 wild American chestnut trees 40 years ago, and they have spread. There are still 4 that are alive, and they have spread. There are literally thousands of seedlings under these trees in his forest, which he says have been planted by blue jays and squirrels. He's a professor emeritus from the University of Vermont. So yeah, there are chestnut trees growing all over the place. There was a tornado that touched down in western Massachusetts, and the tree that came back was American chestnuts. Why? Who knows? But they are coming back on their own. And, people may say it's too slow, it's not fast enough, but, the fact is they are coming back, and people are excited. The people who are working with the wild American chestnuts that are blight tolerant are really excited about the progress.
And where can people go to learn more about the work that you're doing?
They can go to our website, which is globaljusticeecology.org, and you poke around, and you can find information about our work on genetically engineered trees.
Fantastic. Anne Petermann, thanks so much for joining us. Anne is Executive Director of the Global Justice Ecology Project. You can find that at globaljusticeecology.org. She's also the co-founder of the campaign to stop GE trees. We really appreciate you being with us.
Well, I appreciate the opportunity. Thank you so much.
It was fun, and it's something that interests me deeply. Well, I'm John Kiriakou. Thanks for joining us for another episode of Deep Focus. We'll see you next time.